Okay, let's move on to discussion of safety issues that arise in drug development, the problems ones run into, and the means that we try to assess these. We screen for safety for toxicity at various stages along the drug development process. In the HITS lead area, we try to remove problematic functional groups and templates. We assess general cell toxicity, and typically we'll run a variety of selectivity screens to assess affinity for other targets that could lead to off-target effects. In the lead optimization stage, where we examine safety issues such as cytochrome P450 inhibition or induction, the potential formation of reactive intermediates, hERG channel affinity, and mutagenicity. And finally, before nominating a compound for clinical lead selection, typically in vivo safety testing will be done on clinical candidates. Such studies can include single dose to dose ranging in vivo safety studies. Our goal for a drug is to maximize the time that a compound is exposed in the plasma at concentrations above the minimum needed for its biological activity, above the Cmin, but below a toxic level where we might start seeing side effects. And so the idea is to maximize the time you're within what's called the therapeutic window to maximize its biological effect. A non-selective drug can result in a variety of off-target or adverse effects. We showed the example of Seldane's affinity for the hERG channel leading to cardiovascular toxicity. Another classic case is thalidomide. Thalidomide was prescribed as antiemetic to pregnant women in the 1950s. Unfortunately, this led to a series of very severe birth defects, and it was turned out that thalidomide was severely teratogenic. As a result, one of the standard things that's done is screening a diverse set of target classes against our lead compounds, looking for potential issues. Typically, we'll profile a series of enzymes, receptors, ion channels, and transporters, looking for selectivity issues. This is typically done outside at companies such as Serup and Pan Labs. Drug-drug interactions can be a major issue in drug development. Drug-drug interactions are due to changes in CYP cytochrome P450 activity, either by inhibition or induction of the cytochrome P450, affecting the metabolism and clearance of drugs. One drug can inhibit the metabolism of a second drug by blocking the CYP responsible for its metabolism, resulting in that second drug's accumulation, potentially leading to toxic effects. Conversely, a drug can induce the metabolism of the second drug by stimulating its CYP production, resulting in low, possibly subtherapeutic levels of the second drug. Typically, what's done to assess this is to screening your drug in the presence of a standard selective for a particular CYP to assess any potential liabilities. Reactive metabolites are another issue that arises in drug development. Biotransformation of a drug into a reactive metabolite, which can then bind to a protein or nucleic acid, can have toxic effects. There are a variety of screening assays for reactive metabolites. We can directly try metabolite ID. Glutathione trapping assay is another standard assay. Glutathione is an endogenous compound that traps reactive compounds in the body. This can be simply added to a microsomal or hepatocyte incubation with your test compound, and followed by analysis for any glutathione adducts. Covalent protein binding assay is another assay that's used, in this case with a radiolabeled compound, and followed by analysis of the incorporated label. Examples of cytochrome P450 in drug development, we talked about Seldane. Seldane is normally completely metabolized by a CYP3A4 to its oxidative metabolite Allegra, typically leaving no measurable blood levels of Seldane. However, when dosed with another drug which inhibits CYP3A4, Seldane can achieve significant systemic concentrations, leading to its cardiotoxic effect. So if this is the standard route of metabolism for Seldane to Allegra, co-dosing with a CYP3A4 inhibitor like the antifungal ketoconazole blocks the activity of 3A4, thus raising the concentrations of Seldane and leading to, unfortunately, cardiotoxicity. Typically, if you're dealing with a drug-drug interaction due to CYP induction, you might actually just see high rates of metabolism and not toxicity. However, there are cases where induction of the CYP can result in a reactive intermediate with toxic consequences. A classic case is the combination of acetaminophen or Tylenol with the CYP2E1 inducer ethanol. This is the metabolic pathway for acetaminophen. The standard route takes acetaminophen through this reactive intermediate, which is then trapped with glutathione. However, in the presence of ethanol and induction of CYP2E1, we overwhelm the glutathione, leading to protein and nucleic acid adducts, which in turn can lead to hepatotoxicity. And this, unfortunately, happens in cases where you have compromised liver function, such as in alcoholics. Liver failure is not an uncommon result of Tylenol and alcohol combinations. We talked a little bit about the blockade of the hERG potassium channel. hERG is the hERG ether agogo related gene. It's a critical voltage controlled ion channel controlling potassium levels in heart muscle tissue. Drugs which bind and block the hERG channel lengthen the time needed to repolarize cardiac cells, which can lead to arrhythmia and life-threatening ventricular fibrillation. Not all drugs which bind the hERG channel lead to arrhythmia. However, the Seldane case that we've mentioned several times has caused very focused attention by medicinal chemists on this issue. Screening for hERG issues can be done in vitro with a whole cell patch assay on cardiomyocytes. It can be done ex vivo on isolated guinea pig hearts, the Langendorf assay, or in vivo using dog cardiovascular telemetry. As no crystal structure of the hERG channel has been obtained, approaches such as mutagenesis, homology, and pharmacophore models have been used as guides for medicinal chemistry to reduce hERG affinity. And some general rules of thumb have been developed to try to reduce hERG affinity. For example, there are a lot of phenyl groups in the lower channel of the hERG ion channel, and phenyl groups which are actually present in your drug may interact with that through pi-pi stacking, so removal or replacement of these phenyl groups is one strategy for reducing hERG affinity. The hERG channel seems to prefer basic compounds, so modification of the basicity or removal of amino groups is one method for modifying hERG activity. Addition of carboxylic acid groups many times can be beneficial to reducing hERG attraction. And finally, the hERG channel is rather flexible, so a rigid or cyclized template with your drug candidate can sometimes be beneficial to reducing hERG affinity. I give an example here where the introduction of a carboxylic acid group into a lead beta tryptase inhibitor from Aventis was very successful in modifying a fairly potent hERG compound to one that's about 100-fold less potent. Mutagenicity, of course, is a key problem in drug development. Mutagens causing damage to DNA or chromosomes, potentially leading to carcinogenicity. Standard screens for testing for mutagens include metabolic activation to assess any mutagenicity of metabolites that may be formed upon dosage of a drug. Ames test is one of the classic tests for mutagenicity. Others include the micronucleus assay, the COMET assay, and the chromosomal aberration assay. Prior to compound selection and GLP toxicity screening, we'll typically run some preliminary in vivo safety studies to assess lead candidates. These may include an acute single-dose toxicity study, a mini-tox study, which can vary in format, but 5 days of 2 dose levels in a rat is a common format. And finally, range-finding toxicology studies may also be run, looking to obtain both a preliminary safety assessment and also to try to help set doses for your GLP tox study. Standard protocols are a week to 2 weeks in rat and a non-rodent species.